Hi there, I'm Jan Witkowski here at Cold Spring Harbor Laboratory on the occasion of the 85th Cold Spring Harbor Symposium on Quantitative Biology. The topic changes each year, and this year it was biological timekeeping. The symposium has a history of this topic. The first one was way back in 1960, and the previous one was only 14 years ago. And I'm very pleased to have Mick Hastings from the MRC Laboratory for Molecular Biology at Cambridge, England, with me. And I know, Mick, I'm going to mangle this name straight away, but tell us about the suprachiasmatic nucleus. Much easier to say SCN.
I think the TLA three-letter acronym, the SCN. Yeah, the suprachiasmatic nucleus. Well, actually, I think the man and woman in the street, if you say to them they have a body clock that governs when they do things, when they wake up, when they feel energetic, when they're thirsty or hungry, they would agree with us. They don't need a biologist with certificates to tell them that they have a body clock. The bit of detail there is that there's a structure in our brain called the suprachiasmatic nucleus because it sits above the chiasm, which is where the optic nerves cross over at the base of the brain. And that, in fact, is our central body clock. We have it, mice have it. You go down the vertebrate groups, it's there to see. And I'm a bit of a disciple for it. It's because it does so much with so little. There are about 20,000 nerve cells and the supporting cells called astrocytes, which make up this small piece of neural circuitry. And yet that's what makes you and me wake up in the morning, makes us prepare to go to sleep at night, and everything else that follows from that. So yes, it is a tiny bit of a mouthful, but it's a fantastic piece of neurobiological engineering to do so much with so little.
I did wonder about the number of cells it has, because 20,000 in terms of a nervous system is an absolutely minute number of neurons.
It's just a drop. It's a drop in the ocean. And each of those individual cells, we think itself is a cell autonomous clock. But in fact, that's the case for many of our tissues. Even if we take a scraping of skin fibroblasts, from you or me or a mouse or whatever, there's still that intrinsic ability for cells to have this 24-hour timing mechanism. What's really clever about the SCN is that it's able to control all of our tissues, coordinate their activities in time. So our digestive enzymes, the way in which the livers and kidneys deal with metabolic poisons and what have you, all these changes on a regular daily basis. And of course, that adapts us to the world outside, the world that we've evolved to inhabit. And the other strength of the SCN, in addition to being able to control everything downstream from it, is that it's the only part of our circadian, our 24-hour system, that's connected to the light-dark cycle. It receives direct retinal inputs from the eyes, from the retina. So it knows when it's light and when it's dark, and so it can establish a program of our internal physiology, metabolism, and behavior that adapts us to what we expect to experience. And that's what makes us a great biological machine.
So it really is the connection between our internal world and the external world of light and dark.
Yeah, absolutely, definitely.
So how does it exert this control over? It's only got 20 neurons, so it's not sending connections to every part of the body, saying do this or do that. How does it exercise this master regulation?
So I should have said 20,000. If I said 20, that was being an over-disciple.
Sorry, yes, sorry, you did say 20,000.
I've told myself a million times, don't exaggerate. Yeah, 20, that's it. So it sits in a part of the brain called the hypothalamus. And as you'll be aware, that's a more primitive part of the brain. It controls our autonomic nervous system, our endocrine systems, basic moods, our responses to physiological stresses and what have you. So in an engineering sense, the SCN creates a very powerful 24-hour timing signal, and then it plugs that timing signal into its surrounding neural circuitry of the hypothalamus. So in that way, the signal is amplified, it's passed on down through our autonomic nervous system, as I say, through release of hormones by the pituitary and the adrenal glands, so that the signal is amplified at each step of the pathway away from the SCN. But say remarkable engineering.
And you mentioned a moment ago that there are 20, I'll get it right this time, 20,000 neurons in the SCN. But you also mentioned astrocytes. Are there an equivalent number, and what's their function?
So,
do they have any function?
Yeah, so as we've known for a long time, astrocytes are a type of glial cell. That name arose because when they were first identified, they were thought to be a glue to hold the nervous tissue together. And obviously there's truth in that. The astrocytes are there to support neurons in many ways, metabolic support, to control the overspill of neurotransmitters that are released when neurons communicate with each other. So there are lots of maintenance roles of astrocytes right across the nervous system. In the SCN, there's probably about a ratio of 4:1 neurons to astrocytes. There are more neurons than there are astrocytes, but there's a significant number of astrocytes. And we and others have identified that in addition to this supportive role that we'd expect them to play, in fact, astrocytes also have an internal clock mechanism. And if you do genetic manipulations in mice, so that astrocytes in the SCN are the only cells in the animal that has an effective molecular clockwork, that astrocytic clockwork can still control daily cycles of rest and activity in the mouse. Obviously, that means that the astrocytes must be controlling the neurons, because we've already said the time signal gets out from the neurons to the surrounding hypothalamus. So it's not to say that neurons aren't necessary, but what we are saying is that surprisingly, an effective cellular clock in astrocytes is sufficient to make an otherwise clockless mouse rhythmic in its daily behavior patterns. Yeah, it's quite a novel finding.
So why do both astrocytes and neurons, why are both cell types able to exercise this control? I mean, is it just redundancy, or there's some sort of deeper reason for having the two cell types?
I suppose at one level, given that we, as I mentioned earlier, even fibroblasts have got a cell mechanism inside them. So that basic molecular timing system is common right across our tissues. So that's the starting point, that's the template. But the other thing is that we think of it as a sort of yin-yang relationship. I know you're keen on the internal combustion engine. And obviously, I can't remember whether Jensen Interceptor, is it 4-cylinder, 6-cylinder? What engine do you have?
It's a Chrysler 440 big block V8.
And the point is, you know, the engine's really powerful because there's one piston's going up, the other piston's going down with the explosion of the fuel. So to have a mechanism where you've got two active, or in the case there of a motor car engine, 4 active components working in antiphase or quarter phase to each other, it makes it a far more robust system. So that, obviously, I didn't see evolution happen, but I would say that this mechanism, because the astrocytes are active at nighttime when the neurons are not active, and vice versa, the neurons are active in the daytime when the astrocytes are inactive. And that's why we come up with this idea of a sort of yin-yang relationship, that one is driving the other, is driving the other, is driving the other. And when you've got multiple components in there, as you say, there's an element of redundancy arises from that, but it does mean the SCN is able to do what absolutely it needs to do for evolution. It needs to give a completely definitive, high amplitude, very robust time signal that's going to be distributed across the body. So that's our speculation as to how this situation has arisen. But it does mean there's a redundancy in there. So as I say, the experiment I just described, where it's only the astrocytes that have a clock, that can still drive behavior. Equally, if it's only the SCN neurons that still have a clock, that again can still drive behavior. So it's a question of necessity versus sufficiency.
For people watching, I should just give a slight bit of background why the internal combustion engine got
I'm
involved.
sorry. Yeah.
I have a classic English car, a Jensen Interceptor, 1974 Jensen Interceptor. So what happens if you completely shut down the SCN? How long do the intrinsic clocks of the cells sort of keep going in any circadian rhythm?
Yeah. So if one's talking, this is really interesting. So dogma keeps getting overthrown, doesn't it, in science? So let's say 20 years ago, the dogma was the SCN is the one and only clock, apart from something in the pineal gland and something in the retina. But essentially, the SCN is the one and only clock. It directs everything that happens. And then when the field discovered circadian clock genes, and it was possible to look in other tissues and suddenly realize, well, hang on, there's a clock in the liver, there's a clock in kidney cells and fibroblasts, as I say. So then it came one of, oh, well, the SCN is still the important thing. And what it does is that it coordinates everything else across the body. And so in that model, one would expect, in answer to the type of experiment you suggest, is that once you inactivated the SCN and took away that coordinating signal to the rest of the body, all those clocks across the body would start to damp down and run out of phase with each other. Mm-hmm. And at the level of the whole animal, that does happen. So if you're looking at endocrine rhythms, for example, which were beautifully circadian cycles of corticosteroids being high when the animal becomes active and then falling over the course of the active phase, they flatline. So in the aggregate, on average, everything does lose that, that coordinated temporal control. But equally, if you were to take some cells from such an animal, put them in a culture dish, you would see that individually, they still have the ability to oscillate. But of course, there's no more synchronization coming from the SCN. So at the level of the organism, everything flatlines, and circadian physiology and sleep, wake, and what have you is all compromised in circadian control.
So tell us a little bit about the work that you presented at the symposium. And you developed a model about how all these complex networks interact with each other.
So I was in a session on neural clocks, and so the question, and it's not an original question of mine, it's a question of the field. Yeah, why is the SCN such a brilliant clock? And of course, one answer to that is because it has to be, because if it wasn't, then it would disappear from the gene pool, as it were. But the real question is, how is it such an effective clock? And so the talk was partly review, so it's looking at work that other people have contributed to, significant amounts. But the first level analysis is, well, what's the nature of this cell autonomous timing mechanism, which is present in SCN neurons, but it's also present, as I say, in fibroblasts and everything else. And spoke briefly about there's a transcriptional translational feedback loop. So these clock genes are activated and they encode RNAs, which translate into proteins, and then those clock proteins then go into the nucleus and turn off the activation of their cognate gene. So you have a delayed negative feedback loop, and that takes approximately 24 hours in its oscillation. It takes about 12 hours to produce the proteins, and then 12 hours for the proteins to be broken down once gene expression is interrupted, and then it can all start again. And there are several groups of proteins involved in this. So again, going back to that redundancy idea, the system doesn't put all its eggs in one basket. There are interactions between the proteins that confer stability and robustness to that cycle, which is in the cell. But then the next level is to remember that in the SCN, in the neurons of the SCN, of course, they're electrically active in the daytime. And that's the same for a mouse or a human. So it's not related to whether you're diurnal or nocturnal. It's just that in the day, when the circadian system anticipates there's going to be light in the world, that's when the neurons of the SCN are electrically active. And the fact that they're electrically active will cause them to have increased levels of intracellular calcium. And there are some components of the cellular feedback loop that have so-called calcium response elements in the parts of the gene that control their expression. And so what actually is an output of the molecular clock, the increased electrical activity of the neurons, becomes a feedback into the molecular clock, to control those particular components of the feedback loop through these calcium response elements. So again, that adds stability, will amplify the signal, etc. Then the next level to think on is, well, it's 20,000 cells, and it's duplicated on either side of the midline. The morphology of the brain is as a third ventricle, which is central cavity, so there's an SCN on either side. But how do those neurons talk to each other? It's not just, they're not operating in isolation. And a large part of the work that we've done, and also that the field has pursued, is the idea of neuropeptides being important. Now, when we think of neural signaling and synapses, we think of sort of millisecond, really rapid, spatially specific, temporally specific coding of information going from one neuron to another. But the neurons of the SCN express various types of neuropeptides, which are released, we and others have shown they're released in a slow, progressive way. So over the course of the circadian, approximately 24-hour cycle, will be waves of neuropeptide moving through this circuit as a form of time cue from one SCN population to another. That's another level now of integration. If people can bear with me, there's a level beyond that. We can now start thinking about the astrocytes, because so far we only spoke about the neurons, but what we've discovered is, as I say, the astrocytes are active at night when the neurons are inactive, and vice versa. And we think that that's because the astrocytes release a neurotransmitter called, well, glutamate. We're familiar to glutamate as part of the metabolic cycles, but it's also used in the nervous system as a transmitter. The astrocytes increase glutamate levels in the circuit. So at night, that neuropeptide wave has disappeared, and it's replaced by this wave of glutamate. And the consequence of that glutamate is that it suppresses the activity of the neurons. And it's only at the end of night, when the glutamate's no longer being released by the astrocytes, that the neurons can then become active. So again, you've got, I used a bit of jargon of reentrant loops. At each level of those, at each organizational level, something which is the product becomes an input to the cycle. And so you can go from within the cell to the fact that the cell is electrically active, it's a neuron, to the fact that the neurons communicate through neuropeptidergic cues in circadian day, to the fact that then overlying that, there's this astrocytic control in circadian night. And the conjecture is that that's what makes the SCN so fantastically robust. We do experiments with organotypic slices of SCN from genetically modified mice. And as long as you provide them with culture medium and you don't get an infection, those slices will keep oscillating beautiful circadian cycles of gene expression forever, for literally for months, certainly 2 years if you keep it clean.
I was going to ask you why is it necessary to have all these different levels, but you've just answered it, that it lends the system robustness, that you can presumably modify one level without completely destroying everything else that's going on.
Yeah, that's a very good point. That's a very good point. So going back to the idea of these waves of neuropeptides being released in the SCN circuitry, but as I say, there are different populations of SCN neurons releasing different neuropeptides locally, but they also project to the targets in the hypothalamus as well. So exactly, you could have population X releasing peptide Y at one time of day, and population Z doing it with peptide Q at a different. So yeah, you do build into it flexibility and output for sure. Yeah.
So you mentioned that that mammals have an SCN. Do reptiles have an SCN or an equivalent?
Yeah, as one goes down the phylogenetic scale in vertebrates, the SCN remains there, but it's accompanied by a more elaborate network of local clocks. I think the best example for that, in fact, the first one identified in the vertebrate is the pineal gland. It was identified by a chap called Mike Menaker in sparrows. And so in birds, quite often, there's the SCN in the position they expect to find it, because as I say, it sits above the optic nerves, and that's common to all vertebrates. So there's a clock there, but there's also a clock in the pineal gland. And what's really neat, of course, is in lower vertebrates, the pineal gland has its own photoreceptor cells. So there you've got the SCN with its input from the retina, you've got the pineal gland that's intrinsically photoreceptive. So there's a more complex network of interactions between these groups. And equally, the retina in lower vertebrates, the eyes can be very important. They have a local clock, obviously it's entrained by direct lighting cues. So turning the telescope around the other way, what we'd say is that in mammals, the intrinsic ability of cells to be rhythmic has been retained, but the overall control to the system at large has collapsed down, it's focused right down onto the SCN.
And that then leads me to ask about invertebrates, because invertebrates are such a wide variety of organisms. I mean, what does Drosophila do? Does Drosophila have a little group of cells in its brain that, or again, is it distributed via the ganglia that are all over the body?
Well, that's what you say is exactly right. So in the neural clock session, I was preceded by a chap called Michael Rosbash, who was one of the 3 fly people who got the Nobel Prize for circadian clocks. And so he was describing the cell groups in the Drosophila brain, and it's by a rough analogy, you could say that there's a population of about, and the fly people will shout at me now, but this is my take on it. There's about 20, because of course in flies, each cell's identified, it's got a name tag, whereas that's not the case in the SCN. But so let's say there's about 20, give or take, cells, which are really a bit like the SCN. And then they communicate perhaps with another 180 or so cells, which are also part of the broader circadian network, and also control centers in the fly brain that regulate fly sleep and digestive activities and what have you, endocrine release. So you can see parallels in there for sure. Are they the same? Who knows? What's intriguing is that one of the neuropeptides that I mentioned earlier, it's called vasoactive intestinal peptide in mammals, the exact fly homologue of that, pigment dispersing hormone, PDH, or PDF, it's in PDH in crustacea, PDF in flies, I think. But that's exactly the same, it's a homologue, and it's critical in holding that fly clock network together in the same way that VIP is for the SCN. So, that's biology, isn't it?
Right. So I think a final last question. And I guess I probably already know the answer, because I do know that bacterial cells have an autonomous clock. Are there any organisms that do not have a circadian rhythm? I mean, there are obviously sort of clock-like mechanisms or feedback mechanisms controlling various metabolic aspects, but in the sense of being controlled by circadian rhythms.
Yeah. It would be a brave man that said never. I mean, obviously there are other clocks. I started off as a PhD student, as a grad student, working on tidal rhythms in crustaceans. So some organisms, because their environment has a time base of 12.5 hours or lunar cycle, they've also evolved a different timing mechanism. But you're absolutely right. Cyanobacteria are bacteria, but because they're photosynthetic, they have a massive investment in coordinating physiology and metabolism to anticipate a light dark. And they've got the most gorgeous circadian mechanism. obviously its biochemistry is very different from that of mammals, but the principles are the same. Even archaea have circadian metabolic cycles as well. I think the thing is that if you're careful with your ATP as an organism, you contribute to the gene pool down the line. And so having a temporal coordination of behavior that mimics or maps onto the demands and opportunities of the world you inhabit makes you a better machine. You know, it's like going back to Jensen, not that a Jensen inceptor would ever misfire, but if it were to misfire, it would use more petrol, it would use more oil, it wouldn't go as fast. So, and as we know, there's a chap called Carl Johnson's done some beautiful competition assays where if you put two strains of cyanobacteria, because that's a tractable experiment to do, which have got different genetically specified circadian periods, the strain which wins out in a competition assay is the strain which is the better adapted to the light-dark cycle under which you grow them.
So that, yeah, I've already kept you so long, but I'm being converted to being interested in circadian rhythms. So what if these what if questions, what if an organism like, say, a bacterial cell, or what if an organism grew up in an entirely constant environment? Would it develop?
Yeah. So the converse happens. There are people working on blind cave fish, particularly in Central America, for example, these limestone cavern systems. And the deeper down you go, obviously, the darker it becomes. And the animals, the fish at the bottom, which obviously came into the cave, eons ago from the top, have lost, as far as we can see, have lost circadian competence. So if there's no environmental drive there to make it useful to have, then obviously by neglect it will disappear. So we know that. Marine biology, as I say, I did that as a grad student. You have to think about organisms and these deep sea vents. I mean, their energy supply is coming from chemotrophic functions. So it might be that even though, again, they populated that location, having been at the surface where one assumes the ancestors would have had a clock mechanism, perhaps they don't need one down there because their energy supply is independent of time.
I did actually write. Oh, I lost it. There was a lovely quote from actually it was Michael Menaker, who summed up the 2007 meeting, and he talks about the next frontier for this field in a more general way was understanding the relationship between circadian rhythms that enabled organisms to live in their environments. So I guess that's really what we've just talked about.
It is, well, yeah.
That was lovely. Thank you very much, Mick.
Thank you. Thank you.
And I'm sorry if I've kept you much longer than I had intended.
I just say well done, Cold Spring Harbor, for actually going ahead with the meeting, even though it was a virtual meeting. Congratulations to everyone who made it happen.
Thank you very much.
Thank you.